This is the group Now You See Me, where we investigated the role of vitamin C in epigenetics.
Here we are looking at epigenetics, just a brief background about epigenetics. If you take any given person, they have multiple cell types, but if you look at those cell types, they're vastly different. Here's just a picture showing some of the different cell types. And when you look at these different cell types under a microscope, anyone can tell the difference between these cell types, but it is of note that they have identical genomes, but instead they have very largely different transcriptomes, which causes changes in the protein levels.
During our investigation, we did not find any significant correlation between our 16 different variables. However, there was a slight correlation between the length and the number of peaks within the gene body. Next, we calculated PCAs and we charted all the genes on the first two components and could not see any type of clustering on the genes. The first two components only account for about 33% of the overall variability.
The next step we did was clustering. When we did clustering with k-means, we found that we had greatest accuracy or greatest homogeneity when we used 14 clusters. The big thing to note here is that one cluster, the purple cluster, accounted for a very, very large portion of the genes. And when I looked at these genes separately, I found that all of these genes were ones that had almost no 5-HMC peaks. When this was repeated with dbScan, we found a similar cluster. And the cluster here that's green matches the cluster here that's purple. And this cluster was a little over 6,000 genes, and 90% of them were shared in common between the two different methods.
Here we trained and tested each classification method on our ARPE cells and then looked at the accuracy of that. All beat the baseline calculated by the naive Bayes method. The confusion matrices for KNN and SVM methods did not predict a single gene to be downregulated. We believe this is because there were too few downregulated genes within the whole. This was only about 99% of the total population. Here's a visualization of the decision tree for the ARPE dataset. It was surprising and rather disappointing to find that length was our greatest determinant in classifying our data. We had hoped for 5-HMC to play a bigger role. To see if the changes in 5-HMC are consistent across multiple datasets, we trained each machine learning model on each dataset and then tested it on each dataset. These charts compare our results to the naive Bayes calculated accuracies. We found here that cyclic AMP was not successful for training or testing on vitamin C related data.
Overall conclusions: the clustering yields many clusters that are possibly related, that have genes that are possibly related in function or related in mechanism. So this is something we would like to look at with pathway analysis later. Another thing is that the classification was largely unsuccessful. Even though accuracies did beat the baseline for many of them, they couldn't distinguish non-differential genes from downregulated or upregulated. Additionally, the vitamin C versus cyclic AMP could not work, and when you trained on one type and tested on the other, it actually was worse than the baseline. Thank you for watching.